So yeah, today we'll see how a contact map is basically generated and interpreted easily. So a contact map is basically a 2D representation of a 3D structure of a protein, and it shows which residues or atoms are in close proximity to each other in the folded structure. So it's like nothing but like a protein. As you know, the protein contains various amino acids, lysine, alanine, glycine, and all the stuff. So its interaction, it's as it's not usually very uh, peacefully visualized in other structures, in the other tools. So I mean, generally in the 3D format. So basically, you can visualize all these interactions in a 2D format, which is basically very, very easy. Now, these contact maps are typically created by calculating the distances between all pairs of the residues, which these are calculated using the C alpha atom of the protein backbone, and the marking those that fall within a certain distance threshold. It is commonly 6 to 8 angstrom mostly. This is the standard one, which usually it's done. And yeah, and this whole thing, like we are the generating the contact map, it is usually done by CMView tool. So and yeah, so it's basically like this. And context maps are symmetric about the diagonal, as I told. So this is like a 2D thing. And the context maps are symmetric about the diagonal because, like, let's say if residue A is in contact with residue B. So it means the same that residue B is also in contact A, right? So the main diagonal in the contact map is basically represents the contacts between a residue and itself, the residue, like let's say proline, lysine, like this. And you will in any structure of a protein, you will see it's always filled. It's like fully dark, and the contacts near the diagonal, uh, near the diagonal, like here, those represents the local interactions in the protein sequence. Okay, so um, so basically, to know if any chain is alpha, which region is alpha helix, what is beta sheet, what are loops. So there are certain things. So basically, in some protein it's like you will get fully color coded fully. So basically you will directly know which is alpha helix, which is beta sheet. So basically if anything is color coded, like if the color is blue, it means the protein is alpha helix. Then if the color is red, it is beta sheet. And if it is green, though, so it is called as turns fully. Okay, now sometimes it happens that you are not getting the color codings in the map. So if in the contact map, um, if like it's basically the thing that alpha helices appear more thicker band, and those are parallel to the main diagonal. Okay. And beta sheets are basically perpendicular to the main diagonal. And loops and unstructured regions are typically have fewer contacts with the diagonal. So these are basically like here, somewhere in the map. Like I'll show you how it is done. So we will just directly go through the tool. So yeah, so the name is CMView. um, you can download it from the website. You can simply search CMView tool and you will get it. and you need Java JRE for that for this program to get execute. Now you will just get a blank window, just to show you how a color coded things are really in this CMView tool. So we'll just take uh, one random protein UV, let's say, and these chain codes are there. Um, so there are many chains in the protein A, B, C, D, E. Whichever chain you want, you can take it. And contact type, basically, mostly we can to measure the distance between the two residues. So these contact types are there. So like it's like C alpha, C beta, side chain. Then this is all atoms, I guess, and this is backbone, I guess. So yeah, we will take C alpha only. And if you want to put some limits, you can put it here. Otherwise, just let it be like this. And you can see it's like this is the color coded uh, map. So basically, as I told you uh, in the paint, I guess. So this blue region, this this region basically contains alpha helix, red, the yeah, and the red, this red, this red, this red. So this will contain beta sheet, and there is no green turns in here. So that's not here. Yeah, so it's like this. I will show you in some different protein too. We'll go to file, load, online PD. No, I have the file, and yeah, so albumin PDB, and we will take A as the chain. Okay, now you see here, there is no color coded this thing. So we have to see manually only which could be the regions and what those regions represent. So basically, um, so as I told you, first I will tell you how it works. So as you can see, there is one circle type of cursor, and there is my computer's cursor, right? So if you see, let's say I am clicking on this. As you can see here on the um, right bottom corner, there is this residue numbers which are interacting. Okay, so it's like this. So this alanine and methionine, these are in contact, but these are very far from each other, right? So how you can interpret this graph? Okay, so as I told, this diagonal line, this strong diagonal line from like top left to bottom right represents the contact between the residues of the protein, like that are close in the protein sequence, like close in the protein sequence. Okay. Now, this is typical feature of the contact map. Okay. And there is this off diagonal patterns then. So the small dark spots which you are seeing. So what exactly they are, like these small dark spots and this lines of the main diagonal here. Okay, so these are basically nothing but represents the long range interactions between the residues, and which are like far apart in the sequence, but they are close to the 3D structure. So these are like particularly important for the understanding of proteins um, a tertiary structure. Then there are secondary structure elements like the short, thick, parallel lines are there, right? So here it is. Um, so here there is one parallel lines. Okay. So these lines basically represents the alpha helices, and the longer perpendicular to the main diagonal, probably like they indicate the beta shifts. Okay. Now, this basically we have to do because there is no color coding here. So you can basically see which is alpha helix, which is beta sheet, which is beta turn through the pattern which you can see here. Okay, and as as you can see, as I'm moving the cursor, some this the circle type of cursor is also moving, and it is moving diagonally, right? So this is the beauty of this tool, which is of the representing in the 2D format. Like for example, uh, this and this. This is like proline and isoleucine, I guess. So these are these are interacting, but these are very very far from each other. So you can see you can say proline and isoleucine. Um, Mm, uh, Proline solutions are like not interact, like they are having the long range interactions um, in protein. So yeah, basically you can analyze like this, and there are different methods also. Like there are these different selection modes also, like square selection, fill selection, diagonal selection. Like I'll show you like so selection in the sense you can drag it, you can so if you have any region of interest, you can select it, you can delete it, you can see this is delete selected context, so you can delete it directly. And the region which you don't want in your protein, you can do it through this thing now. And there is one more feature in this. um, there is this compare. Okay, so what you will do is, um, so this compare will I will show you directly instead of just telling. uh, so we'll put again our this protein only, and instead of um, giving a chain, I will, let's say we'll give L chain. Okay, now you see. And yeah, and there will be a sort of alignment between the chain A and chain L of this protein. And this is done by this Needleman Wunsch sequence alignment. This is one of the matrix alignments which does the alignment of two proteins. And you see. So now what is happening? These two proteins chains are aligned on each other. So what exactly it does is, I will tell you. Um, so after our second contact map has been loaded of the chain L, so as you can see here, this black contacts here. So this black contacts represents like these are the contacts present in both the structure, like the chain A and chain L. The second one is the pink, as you can see here, everything here, like in this region, full pink is there. So this pink contacts present in this contact map A, like the A means our um, chain A and the chain L. So it means that the contacts are present in chain A, but those are absent in the chain L. So like this, again, there is this green type of color. So this represents that the contacts present in the chain um, L, the contacts are present in chain L, but they are absent in chain A. So yeah, you can compare two different types of proteins also here. There will be some case also that two proteins won't even match, and every region, full this full region will get green in color. So I can we can do it, but the video will be very very big. So basically, what happens is if let's say if there is any protein, let's say insulin, okay, and there is another protein I load, which is let's say of GABA receptor of the for the neurotransmitters, right? So these two proteins don't have any match or anything with each other, right? So all the residues, like whole region will be green in color because both the context present in both the proteins doesn't even match with each other, right? So yeah, so like this we can do. There are some overlays here, like this bottomless beachy difference map is there. So yeah, you can explore this one. There are more. The you know Remember the first protein which we loaded in this tool. There are more overlays. This distance map is there. Like what is? Uh, oh yeah, you can see this diagonal architecture of this thing. This is the matrix behavior. Um, this red regions represents high density contacts. Okay. Uh, the blue represents there is nothing in the matrix. If there is, it's of no use. So yeah, you can explore this tool like this, and means yeah. So all it's I guess all done through it. This run peel. There are some algorithms also based on some tool, but I'm not sure about this. All this. Yeah, it's something like a Needleman Whoosh algorithm again for the alignment purpose. So yeah, it's all about um, this tool. There are more things to do with this show difference map, and like there are so much things from so much things to explore in this. You can even connect it to the PyMol. Um, you can um, you can select two, three. Um, you can select the your protein of the region of interest, and it will connect to the PyMol, and it will show in which region this which where is this selected region in the 3D format. So yeah, there are so many things to do with this. Um, yeah, and I don't think so. There is much more to tell about this tool. Uh, Still, yeah, and there is this one show loop. So if uh, it just gives slight good view of the matrix, uh, and according to your region of interest, you can select 1111 residue from it. So yeah, it's all about this tool. Yeah, and yeah, see you in next video. Thank you for watching.